Welcome everyone to this webinar on the regulatory acceptance and use of next-generation approaches for chemical safety assessment, where we'll discuss how the most advanced methods in toxicology are being deployed to ensure the safety of chemicals. Today, we'll hear from our speakers on how industry ensures product safety through the use of non-animal approaches and how the Government of Canada is using the latest technology based on non-animal methods in chemical management. We have with us today Dr. Julia Fenton from Unilever and Dr. Tara Barton-McLaren from Health Canada. We'll first hear from Dr. Fenton, who leads Unilever's team of safety and sustainability scientists. She has been active in the development, validation, and use of non-animal approaches for assessing chemical safety for over 30 years, initially working for an animal protection organization and then the European Commission before joining Unilever in 1998. Looking forward for this presentation. To you, Dr. Julia.
Hello, everyone, and thank you, Marina, for the opportunity to share some of our work in using new non-animal approaches for assessing the safety of the ingredients that Unilever uses. We now have over 50 safety scientists in SIAC actively applying next-gen tools and using data from NAM's new approach methods for safety assessments. So firstly, I would like to acknowledge their contributions and especially thought leadership from the science leads listed here, in partnership with our many scientific collaborators. We are over 2 decades into the 21st century, so it makes sense that we are using 21st century science and technology to make decisions on chemical safety. The scientific partners we have worked most closely with on this journey are shown here. Over 70 of them across the world, all committed to developing new safety science approaches which enable us to avoid having to do animal tests for assessing the safety of ingredients used in consumer products. My role, I am accountable for the scientific capability and assessments that ensure Unilever's products are designed to be safe for consumers and our environment, and that the processes in our factories are designed to protect our workers from harm. It's essential that the decisions we make on chemical safety are based on the best scientific data and knowledge available. Over 3.4 billion people a day use a Unilever product. Rapid advances in biology, in analytical and computational approaches, and in cell-based technologies and assays have led to new types of more relevant scientific data being generated to improve the safety decisions we make, whether that's the safety of consumers, workers, or citizens exposed to chemicals in the environment. New approach methods or next-gen tools provide different data inputs for exposure, hazard, and safety risk assessment based on a much more detailed understanding of the relevant biology and chemistry. About 20 years ago, EU policy and proposed legislative changes concerning animal testing of ingredients used in cosmetics stimulated the scientific community to rethink and transform our approach to assessing consumer safety. A wide range of scientific approaches are now used routinely to generate the data needed for the safety assessment, taking a tiered approach tailored to the specific safety questions being asked. For example, are we concerned about dermal effects, potential harm from inhalation, or the possibility that an ingredient may penetrate the skin so there is systemic exposure? Some of these methods are now standardized and accepted as testing guidelines, but in some cases, there is a need for much more complex safety assessments, adopting so-called next generation risk assessment approaches. And these are what I will focus on in the rest of this presentation. So next generation risk assessment is defined as an exposure-led, hypothesis-driven risk assessment approach that integrates new approach methodologies to assure safety without the use of animal testing. It draws on leading-edge expertise in biology, chemistry, risk assessment, informatics, modeling, and various other scientific disciplines. It's team science focused on generating the key data needed for making safety decisions. Under the EU REACH chemicals legislation, we have to comply with the principle of any animal testing being as a last resort. So it is these NAMs and next-gen approaches which we are now starting to use to meet this requirement. An outcome of research funded by the EU under the SURAC programme was this tiered approach for using a wide range of NAMs for assessing the effects of chemicals on human health. And the next-gen tools required have been further developed under subsequent EU research programmes, such as EU Tox Risk. Currently, they are being applied in case studies, involving many scientists collaborating across industry, academia, and regulatory teams to help define standard workflows and develop guidance. Together with the excellent work done on adverse outcome pathways under the auspices of the OECD, the implementation of IATA, integrated assessment and testing approaches by the Canadian and other authorities, and the work led by the US EPA to implement the US National Academy of Science 2007 toxicity testing in the 21st century strategy. This huge amount of scientific evidence and expertise starting to provide confidence that we can assess the safety of chemicals and protect human health and the environment by using these new types of data and tools in this type of tiered approach. Whilst the scientific community working on NAMs is confident that we now have better and faster tools and data for chemicals management, we are again facing the same challenge that I battled against when I was working for the European Commission Joint Research Centre, a TechFam, over 25 years ago, when we were trying to replace skin and eye corrosion and irritation rabbit tests with validated non-animal tests and testing strategies. Animal tests underpin the chemicals regulations. Despite all the uncertainties in how relevant the animal data are to taking safety decisions that protect people and our environment from the harmful effects of chemicals, and despite the fact that EU REACH chemicals regulation was implemented 15 years ago to promote alternatives to animal testing as a better way to protect human health and the environment. NGRA approaches are anchored in detailed understanding of the relevant biology and exposures. They do not attempt to predict the results of high-dose toxicology studies in laboratory animals. Why would we? Given that those animal studies themselves are at best 50% predictive of adverse human health effects, albeit they are still mainly protective of human health due to the various uncertainty factors that are applied to the animal data during the overall chemicals safety assessment and risk management process. Our NAMS toolbox enables us to investigate how chemicals interact with a wide range of biological pathways and activities at relevant exposures. To use these data to make safety decisions, some key requirements were, firstly, an understanding of the biokinetics to be able to link in vitro and in vivo exposures. Secondly, approaches to integrate different types of diverse data in a consistent and transparent way. And thirdly, a relatively simple and standard metric to represent the margin of safety, or the so-called point of departure. This is known as the bioactivity exposure ratio, a critical unlock really for the use of NAMs for chemical safety decision-making, and described in the excellent paper from Katie Paul Friedman, which includes leading regulatory scientists from US EPA, from the EU, ECHA and JRC, and from Health Canada as co-authors. As one of these co-authors, Tara is better placed than I am to talk more about this in her presentation. When we are interested in understanding how chemical exposures may cause systemic effects, our key tools are biokinetic, PBK, modeling, and other exposure tools, high-throughput transcriptomics, the cellular stress, and in vitro pharmacological profiling. We are essentially repurposing in vitro tools and computational modeling approaches used in new molecule discovery and assessment in the pharma and agrochemical sectors, and bringing the same modern science to bear in improving how we assess the safety of more general, less bioactive chemicals. For our work on ensuring consumer safety of chemical ingredients, understanding of the exposure kinetics is essential to being able to develop and document the integrated scientific risk assessment, with the plasma max concentration being a key data input for calculating the bioactivity exposure ratio. We have now defined and published the frameworks with some supporting case studies that we are using to ensure we apply NAMs in a consistent and transparent way for assessing consumer safety. Our work on skin sensitization and systemic exposure is the most advanced, and on these we are collaborating with US agencies to further evaluate the broader applicability of the NAMs for chemical safety decisions. The work that Alistair Middleton has led, a collaboration across the Unilever safety scientists, Charles River and Cyprotex, to evaluate the protective ability of the NAMs toolbox for systemic safety assessment, will be published in Toxicological Sciences shortly. The workflow shows how the outputs from the exposure PBK modeling module and the cell stress panel, high-throughput transcriptomics, and in vitro pharmacological profiling data used to estimate the ports of departure are then combined to estimate the bioactivity exposure ratio. Paper reports promising data for 25 different exposure scenarios covering 11 chemicals as a proof of concept study, enabling a prototype decision model to be established, which will then be used in a much larger evaluation of the toolbox. Now, moving briefly to ensuring worker safety, which is ensured via a prevention and protection strategy. Exposure-based risk assessment to protect workers from chemical exposures is central to all health and safety at work legislation. In Europe, some of the current data inputs on chemical hazards are from animal testing conducted to comply with the REACH chemicals regulations. So again, here the question becomes, are there now better, more scientifically relevant data inputs that could be used so that animal testing is avoided? There's a long history of using PBK modeling for occupationally important industrial chemicals of concern. So it should be feasible to repurpose NAMs with a bioactivity exposure ratio approach for assessing worker safety, as we are doing for consumer safety. We should certainly be consistent in the tools and data we use to assess impacts on human health for both consumers and workers, whilst meeting the EU legislative requirements that any animal testing can be shown to be as a last resort. So if we're required to use NAMs to avoid animal testing for chemicals used in cosmetics, where actually the consumer exposures typically exceed those of workers in factories by some considerable margin, providing, of course, that the proper health and safety controls and prevention measures identified in the local risk assessment are in place. If that's true, then we should likewise find a way to enable the use of robust scientific data from those NAMs in place of animal testing for worker safety. When considering the data needed to protect people and non-human species if exposed to chemicals in the environment, the EU REACH and other chemicals regulations have not kept pace with the rapid advances in science and technology. To afford the best protection, and the best scientific tools and safety data need to be used. So we have gaps to close and an urgent need to build greater capability, capacity, and confidence in the use of NAMs for assessing chemical safety in a regulatory context. And Tara will show how Health Canada is approaching this in her presentation. NGRA using NAMs has been increasingly established for assessing the safety of chemical ingredients used in cosmetics during the past decade, and is now written into the March 2021 Notes of Guidance from the EU Scientific Committee on Consumer Safety. And learning from the scientific and regulatory progress being made under the US EPA's NAMs work plan, various activities are ongoing to try to embed the use of NAMs for chemical safety assessment within the current EU REACH regulations. And there's an example there of a framework developed under the auspices of ESITOC for chemical safety assessment incorporating NAMs within REACH. The EU's chemical strategy for sustainability sets out a more ambitious future trajectory than the current EU REACH and classification, labelling and packaging revisions. For the sustainable innovation strategy to be successful, it must be anchored in advanced science and technology, including modern safety science and the next-gen toolbox. A year ago, some of Unilever's NGRA science leads were invited to share our thoughts on what is needed to transform EU chemicals regulations and why. As intended, our paper provoked some much needed discussions and also ongoing constructive dialogue between the scientific NAMs and chemical regulators communities, such as that happening under the auspices of EPAA. In recent months, for example, at the Helsinki Chemicals Forum, we see evidence of more openness to change and to finding ways to embed NAM-based safety approaches for chemicals regulation. The EU member states have recently shared information on the PARC programme, which should help build the necessary capability and expertise in the regulatory community. So I'm very positive about stakeholder engagement and progressive thinking. That said, we do need more urgency and a clear roadmap to transition to safer and more sustainable chemicals and innovation, grasping the opportunities to change the data inputs and tools used so they are more scientifically relevant than animal testing and afford stronger protection of human health and our environment. In closing, I strongly recommend taking a close look at the data in the 2020 paper on in vitro bioactivity from Katie Paul Friedman and her regulatory science collaborators that I mentioned earlier. Their evidence shows how NAMs used in an NGRA context are both better science and protective, and many are already useful for assessing chemical safety for regulatory purposes today.
Thank you. Thank you so much, Julia. And now we'll hear from Dr. Barton McLaren. She's the research manager of the Emerging Approaches Unit of Health Canada's Existing Substances Risk Assessment Bureau. She has been contributing to human health risk assessments and methods development under Canada's Chemicals Management Plan since 2007, following the completion of her PhD in reproductive toxicology from McGill University. She serves as the focal point for the development of new approach methods and strategies for the assessment of chemicals existing in the Canadian marketplace and contributes to international initiatives such as the OECD and NCRA to promote alignment for the use of modern toxicology and emerging technologies in regulatory applications. Thank you so much. Let's hear from Dr. Barton McLaren now.
Okay, thank you for that kind introduction, and thank you to you, Marina, and to others for the opportunity to share the work that we're doing at Health Canada in the bioactivity exposure ratio context. So if I can just confirm everybody is seeing my slides, that would be great. Get a head nod from you, Marina. The slides are good? Yeah, great. Okay, so I'm going to jump right into it. And I'm going to start just by giving a little bit of context. I know we have a very international group here today, which I'm very, very pleased to see, and kind of give a little bit of a position to where we are at Canada in the context of our work. So the risk assessments of the industrial chemicals in the program that I work in is mandated, or the legislation under which we assess these chemicals, is the Canadian Environmental Protection Act, or CEPA. And CEPA provides the framework for everything that we do, essentially the identification, prioritization, and assessment of new and existing substances. It also is the legislation under which we can control or manage those that are considered to pose a risk as needed. This framework is broad, open, transparent, and evidence-based. And importantly, it takes into account aspects of both exposure and effects, so that the exposure and toxicity, or a risk-based approach. CEPA has undergone review in the recent past, and importantly, some of you may be aware of Bill S5, which is ongoing. And this, in fact, is an act to amend CEPA, or Strengthening Environmental Protection for a Healthier Canada Act. And this Bill S5 came shortly after the introduction of the Prime Minister of Canada's mandate letter to our Minister of Health, which outlined a number of priorities for our Minister of Health. But I just wanted to highlight within those many priorities, to protect Canadians from harmful chemicals, there was also the requirement to introduce legislation to end testing on animals. So as you can see within Bill S5, one of the key themes is, in fact, working toward reducing reliance on animal testing. And if you're interested, I've provided links to both of those. Now, they're looking across the different types of chemicals that we assess and regulate in Canada. I just provide this chart really as an example to see how the requirements for toxicity studies differ across different program areas. So you can see here for pesticides, there is significant requirements for animal toxicology studies at this time. In new substances, depending on the volume of the chemical or the schedule under which the chemical is submitted for assessment, there are moderate data requirements. However, in the program that I work for existing substances, there are no data requirements, and all of the assessments that we conduct are based on publicly available information. And so there are opportunities for new approach methods to effectively support risk assessment across all program areas, but the way in which we introduce them is dependent on the requirements. What's important here is that there are many common goals, that to protect human health and the environment, to work towards reduction, replacement, and refinement of approaches to animal testing, to promote acceptance of NAM and fit for purpose uses. And we're doing this all in partnerships and collaborations to ensure alignment, not only across program areas at Health Canada, but also internationally, and doing this through the building of case examples to work towards establishing best practices and hopefully guidance through the OECD. But ultimately, we're looking to these new approach methods to optimize the pace, the accuracy, and the efficiency of our risk assessment activities. And just as Dr. Fenton very nicely highlighted, partnerships and engagement are absolutely critical as we move towards this risk assessment modernization. And I've highlighted here the various groups that we work with internally, regulatory research groups across different program areas, but also with a number of universities, University of Ottawa, McGill, and international institutes under APCRA and the OECD and others. So it is absolutely critical. I only have a small team of about 8 individuals and scientists in the group. And so really all of the advancement that we're doing is based on the successful regulatory research partnerships and engagement. So what we have been focusing on really is exploring computational and testing strategies, as well as a number of different technologies across the levels of biological organization to develop interpretation and application approaches so that we can better understand the data in different regulatory use contexts. And so this slide illustrates some key areas of ongoing development and focus. And just really in the interest of time, this is just a high-level snapshot. And in each area, there are many studies or many case examples ongoing. But what I'm going to focus on today is 2 examples of where we're looking to use high throughput or high content cell-based assays in order to derive either protective and or predictive approaches so that we can compare in vitro points to departure to human exposure estimates, or this bioactivity exposure ratio. And so we've been using case studies to translate these findings into application. We use the case studies to demonstrate the methodology through research or build confidence in the science. We then refine these techniques into documents, and these documents at Health Canada we call science approach documents, to create usable workflows and demonstrate the best practices for the established methodology. Now, a science approach document describes the novel approach to evaluate the environmental or human health risk of substances. It doesn't make a conclusion under SEPA, but rather it demonstrates the approach that can be used in future risk assessments or prioritization exercises. So then, once we establish the science, create the guidelines or approach to effectively implement the scientific methods, then we can move towards putting that guidance into practice for a variety of regulatory means. And when we're thinking about the in vitro data and the case study findings to establish human relevant endpoints, we really think about this central workflow and common themes working towards establishing the molecular points of departure from the high throughput or the high content data. So first we start with the data collection, and we want to ensure that the design that's used really best describes the potential hazards. There's a lot to think about when we go through the processing of the information and converting the raw values into the usable endpoints. There's certainly a focus on the analysis and interpretation. We need to evaluate different means of obtaining the optimal metric at the end of the evaluation or interpretation in order to derive this molecular point of departure. And then when you're using in vitro data, it's really important to consider the information that's needed to convert the in vitro data into an in vivo or a human relevant value. And then there's also the considerations regarding the application and being able to incorporate these values into regulatory frameworks. So as the first example, I'm going to come back to this foundational paper that was a significant collaborative effort conducted under the Accelerating the Pace of Chemical Risk Assessment, very nicely led by Dr. Katie Paul Freeman. And so this really served as that foundational piece of work where we looked at hundreds of chemical, asking the question of whether or not in vitro point of departure can serve as a protective surrogate in the absence of traditional toxicological data. And so this case study considered high throughput in vitro bioactivity data from ToxCast together with high throughput toxicogenomics modeling to derive these in vitro points of departure. So very briefly, what was the approach that was taken to better understand or address this question? So we used the US EPA ToxCast TOX21 data, and these assays represent targets and pathways associated with toxicity. The 5th percentile AC50 values from all assays meeting a predetermined criteria were extracted. They were converted then using the HTTK R-based package so that we could get milligram per kilogram body weight per day metrics. And then the 5th percentile of all outcomes from a specific chemical were used to derive this bioactivity point of departure. And the figure on the right, I understand, is very busy, but intentionally so, showing that we looked at many chemicals, and for the vast majority of the chemicals, the in vitro values from the ToxCast represents a lower or a more conservative endpoint when we compare them to the PODs derived from the in vivo data. Now at Health Canada, we wanted to better understand what this meant to us. So we took this high level analysis using many chemicals, and we brought it into a scope that was directly comparable to our traditional risk assessments. And so the figure on the right really shows how the BER approach nicely mirrors the traditional risk assessment steps or considerations. And what we did in this science approach document was we looked specifically at about 42 chemicals for which the bioactivity points of departure were available and that we had already concluded on under SEPA. and we extracted the points of departure that were used for risk characterization in our risk assessments, compared those to the bioactivity points of departure, and then also compared both the bioactivity PODs and the traditional PODs to Canadian exposure values, again, that were taken from the traditional risk assessments and included biomonitoring data, environmental media, and consumer products, so that we could compare the BER and the margin of exposure, which is our traditional metric that we make decisions with. And so what we found was very promising. Again, using this approach, all 6 non-genotoxic compounds, and these are in the larger or bolded names of the compounds on the left-hand side of the figure, that were considered toxic to human health under CEPA section 64C, had log 10 BERs that were less than 2. And so these were equivalent to margin of exposure of 100 or less. And so these all fell into the moderate or high concern. And a similar observation was found with those substances that were considered ecotoxic. Now, during this evaluation, we also looked for exceptions. And so there are exceptions, and that's part of gaining experience, is better understanding the space and chemistries for which certain approaches may not apply. And in this case, quinoline was an exception. It is a genotoxic substance. It was both section 64A and C, so eco and human health toxic, and it ended up in the low concern bin. So why? We looked at this more closely, and really this prompted us to continue to explore more in depth an approach that could be used to identify genotoxic substance. And so this study has been nicely led by Dr. Mark Beal at Health Canada, and they've been exploring in vitro genotoxicity data to estimate points of departure. This has been a significant collaborative effort by the IVIVE subgroup under the HESI Genetic Toxicology Technical Committee. And without going into any significant details, essentially they're using the same workflow, in this case, looking at some reference chemicals and applying the GeneTox21 platform to evaluate whether a similar approach could also be applied using, the right assays for genotoxicity. And so here, as kind of a summary outcome, they are also finding that genotoxicity data coupled with IVIVE models can provide surrogate points of departure that are protective, again, of human health for most chemicals. For those chemicals where it is not protective, when you start to dig deeper into those chemistries, the biochemistry can explain most of those results. So looking to better define where the exceptions or uncertainties or even cautions lie in some data sets. And in this case, this multi-assay assessment, again, provides mechanistic insight into genotoxic mode of action for many of these or all of these chemicals. So for both of these examples, there's ongoing work to further refine the approach, acknowledging that there are some limitations when we're using non-animal data, such as looking more into the impact of metabolic activation, as well as toxicokinetics properties. In some cases, we require higher tiered models to be applied, for example. But at any rate, this approach allows for screening and potency ranking of chemicals with a human dose context, which is very important. And it helps us to establish a framework for interpretation of the in vitro NAM-based concentration response data and put that into the context of quantitative risk assessment. I wanted to highlight here, I'm not going to go into the details of this study, but also to acknowledge that when we want to look forward and move forward, especially for chemicals that are data poor, often we don't have the specific data that we would like to have or need to have to be able to derive the BERs or apply the approach. And so we've been looking on building on the EPA generalized read-across approach and using computational models to be able to better address to better address those data gaps when they exist for a broader chemical space. And so here we're looking at using bioactivity read across, as well as different predictive models to fill the gaps for the toxicogenetics data so that we can expand this approach in a prioritization context. Importantly, we acknowledge that as the science advances, we want to be able to refine and add different types of data to these types of workflows. So they've been designed in a modular and flexible way so that we continue to update and use the best available science at the time of application. Okay, so jumping into the transcriptomics data. So we also want to gain experience across different technologies. So here we're looking at measuring changes in gene expression, again, establishing metrics using benchmark concentration models. And for this case study, emphasizing the high throughput nature of the approach and incorporating human relevant aspects using HTTK models. So very similar workflow, but using a different technology. And this again is a collaborative effort. And this case study has been submitted as an IATA to support screening and assessment of endocrine active substances under the OECD IATA Case Studies project. It's under final review, so hopefully it will be available to everybody very soon. So first, what was the regulatory context for this work? So in 2017, 2018, under our assessment priorities review, a subgroup of bisphenols were identified as future priorities. And so the starting point for problem formulation were 31 structurally similar and 8 functionally similar substances. Within our program, we applied a computational approach to identify a broader set of substances for further exploration. And so we actually ended up kind of looking at a number of different inventories and growing the overall subgroup to around 340 unique chemicals, which we then brought back down to identify those most similar to bisphenol A. And so for the goal of this IATA was really to better understand the hazard for this subset of BPA analogs, which are otherwise data poor, and use this as an example of implementation of NAM in risk assessment activities. For this case study, we used MCF7 cells and exposed them to 25 target chemicals, 18 with the bisphenol substructure, and another 7 that had the potential for use as a functional alternative, so a different structure to BPA. We collected, so this is an IATA, so we wanted to look at and compare a number of different types of information. So we collected the in vitro high throughput screening data. We generated the in vitro high throughput transcriptomics data, and we also incorporated in silico predictions. For this experiment, we wanted to look to see how the same technology can be applied for the derivation of molecular-based points of departure in 2 different ways. The first being to derive minimal bioactivity concentration applicable for general systemic toxicity. So thinking about this in a hazard independent way. If you don't know the mode of action, how could we use this data? And then secondly, to derive a minimal bioactivity concentration applicable for ER activation. In this case, we were able to use the ER biomarker to look at ER pathway specific points of departure. Again, we apply the IVIVE to extrapolate from in vitro to in vivo. Not in the case study, but the ultimate goal then would be to compare these to exposure metrics for that VER. So this is the workflow. I think many are familiar that transcriptomics creates a huge data set. We then needed to think about what parameters needed to be applied to identify the genes with a concentration response, and think about a number of different ways to derive the optimal point of departure. And so we did use a number of different ways to be able to evaluate and work towards establishing what might be considered the most robust or the optimal metric. So we used several approaches and first looked to see whether they had agreement with the benchmark concentrations that were derived using the ToxCast AC50 values. And you can see here that among the specific approaches, the 25th ranked genes seem to provide the most robust data, as it did have good agreement with the endpoints derived from the ToxCast data set. And through this evaluation, we also found that there were a number of substances that exhibit the pathway specific activity, and that also demonstrated to have endocrine disrupting properties. So we did many different analyses, but I'm just going to kind of bring this all together and show how we can think about putting the different types of data inputs into a display that helps us better understand the consistency and comparison across types of data within chemicals, as well as across the different chemicals that we're looking at in this group. And so on the left-hand side, where the chemicals are listed, you can see that those are considered to be ER active based on the weight of evidence from in silico models are listed in red. So we see not all were endocrine active based on in silico. We then were able to compare the NAM-based points of departure, which are circled in green. And here I'm looking at or showing you both the ER pathway-based, which is blue, and the general systemic points of departure, which is more in the purple. And interestingly, what we can see here is that the general toxicity and the pathway-derived points of departure, again, are in good agreement. But the value in having generated both of those is in particular for those that did not have a point of departure based on the ER biomarker. So we're able to assess those that may not be endocrine active, but still put them into context in terms of relative potency among this broader group. And then when you compare them to the apical points of departure, which we extracted from developmental, repeat dose, and reproductive toxicity studies, again, these NAM-based points of departure are lower than the animal-based PODs, indicating again that they are protective. So using in vitro models are not without challenges, but this also presents us with a number of opportunities. The first opportunity is really to think about and influence the design of the study so that we ensure we're using the right models. We have ideal exposures and platforms in place. There's a lot of processing considerations, so looking at bioinformatics pipelines, and ensuring that we're able to remove the noise, the outliers, unresponsive data, for example, and that we can optimize the processing of the data. In the analysis, we want to ensure we're using the optimal means or models to capture the concentration response, and that we're using the right metrics to define the optimal points of departure. So a lot of ongoing work to work towards kind of that best practice for the analysis and interpretation. And then again, very importantly, looking at the software capabilities and model assumptions and refinements that go into the IV-IV extrapolations. And then getting to the application where we're using relevant values and being able to include meaningful outcomes such as the BER approach. So bringing this all together, I've showed you a number of examples where we're working really to adapt and demonstrate the application, to innovate and accelerate and leverage and integrate many technological advances towards that acceptance, and where we're working to apply it in priority setting and the ability to identify chemicals and issues of greatest potential concern. Now, we want to acknowledge that there are many foundational or at least 3 key that I've listed here, foundational elements that we continue to work on building and putting in place, from the engagement, the infrastructures, and being able to access and share data more openly, so that we can ensure this robust principles of risk assessment, working towards science excellence or maintaining the science excellence in risk assessment to achieve the desired outcome, to prevent exposures and particularly to protect human health and the environment. So the take homes, really, I think to leave you with, the time is now to innovate and develop the fit for purpose approaches to increase our efficiencies and improve protection. Key international regulatory jurisdictions are committed to the reduction and working towards the elimination of animal testing. And these are pivotal data sources for risk assessment. So we really need to keep pace and align internationally. Research regulatory collaborations are absolutely imperative. And we need to continue to build this common vision and commitment to advance alternative methods and maintain the excellence in science-based decision-making. So many people involved, and want to acknowledge all of those and others within all of these collaborations. And I will end there. So thank you.
Well, Thank you so much. And I would like to thank both our speakers for really great presentations, showcasing what can be done already with innovative non-animal approaches to ensure the safety of chemicals and products. I'd also like to thank all of our attendees for taking the time to join us, and I hope you found this session useful and insightful.